Hello, I'm Sally, and today we're speaking with the wonderful Professor Omer Aziz and Professor Patrick Caswell about PMP and appendiceal tumours. Coming to you, Omer, I'm wondering if you could please introduce yourself first of all.
Sure. My name's Omer Aziz. I'm a surgeon based at the Christie Hospital in Manchester, and I'm a researcher based at the University of Manchester and Manchester Cancer Research Centre. And my area of interest is tumours of the appendix that result in rare conditions such as Pseudomyxoma, peritonei, or PMP.
Amazing, great stuff. I'm coming to you, Pat, next. I'm wondering if you could just give a brief overview of who you are and what you do.
Sure. I'm Pat Caswell. I'm a professor of cancer cell biology at the University of Manchester. So I'm a basic researcher. I'm interested in fundamental mechanisms, in particular how different cancers spread in the peritoneal cavity, the body cavity, and how they survive there and then reach distant sites from there.
Great stuff. And just coming into appendix tumours, could you tell us a bit about their incidence and kind of, you've talked about this PMP and the peritoneal metastasis. How does that happen in reference to the appendix tumours?
So appendix tumours are rare. They happen in less than 1 in a million people. They range from being low-grade appendiceal mucinous neoplasms. So these are tumours that might take 3, 5, 10 years to develop into a condition where that tumour spreads into the tummy lining, and that's a condition called PMP or Pseudomyxoma peritonei. So that's one range. And on the other extreme, you have appendix tumours that behave very much like bowel cancer does, and they are more aggressive, and they're called adenocarcinomas. And they may spread not just to the lining, but also to the other parts of the body. They tend to present a little bit sooner, so maybe 2 to 3 years after they develop.
Okay, thank you for that overview. I'm just wondering if you could talk a bit about the type of cancers that you research within your team.
Yeah, so we are interested in tumours of the appendix. So the appendix is a part of your body that people don't really know what the use is for in a human. And these tumours range from low-grade, slower-growing tumours, which are appendiceal mucinous neoplasms, they're called, to the more aggressive type, which can spread to other parts of the body. And again, they're adenocarcinomas, like bowel cancers, or rarer tumours called goblet cell adenocarcinomas.
Amazing. And what is the kind of rarity of this cancer? What's the incidence?
So each of these types of tumours occurs in around 1 in a million people. So, in a 70 million population, there'd be 70 new cases. And so for each of these types of tumours, there are probably, 1 or 2,000 people walking around with that tumour in their appendix.
So you've mentioned that this is a rare cancer. What does that mean for treatment options and patient outcomes?
So because it's a rare cancer, there are only 2 hospitals in the country really that specialize in tumours of the appendix of this type, and we're one of them. So patients will present with these tumours quite late. They don't tend to feel any symptoms from them, and the way they often will present is when the tumour has spread into the abdomen. They might get abdominal swelling or pain or discomfort. And so the treatments generally involve surgery, and it's surgery for very late and advanced tumours.
My next question would be, with all that in mind, what does the research here in Manchester focus on in regards to this cancer type?
Because these tumours present late and we haven't had that many of them, there really aren't any new treatments that have been used towards them. And by that, I mean systemic anti-cancer treatments like chemotherapy, for example. We use drugs which are outdated in other types of cancer, but because we haven't been able to study the types of drugs we could use for this cancer because of its rarity, we have to develop a way of testing new drugs, identifying targets, without having lots and lots of patients with the disease. And that's been our focus in Manchester, is to try and create what are, models which aren't in humans, from which we can understand the tumour and then figure out what kind of drugs we could use for either advanced disease or early disease.
Okay, so what you're saying in this is that there's not presently any standard of care anti-cancer treatment for this type of cancer.
Absolutely. So the types of treatment that currently exist extrapolate from colorectal cancer, bowel cancer, into these types of appendix tumour. And so we can't really predict whether patients are or aren't going to respond. It's a bit of a lottery.
I'm wondering if you could kind of summarise what you've said there in terms of what are some of the key challenges in treating this kind of cancer.
So one of the key challenges is that with very late disease, patients have tumour on a number of organs in their abdomen. And so that can mean quite dramatic surgery for them. It can mean that they need bowel resections. It can mean they need important structures like ovaries and the uterus removing in women. And that means that the surgery they have can be very life-changing. So the biggest challenge in appendix tumours is this disease presenting late and us not having anything to melt the tumour away that could save those organs.
Incredible. So I guess looking to the future and what we're doing here in Manchester, Pat, you're leading a really interesting study on the establishment of organoids. Please could you tell me a bit more about this study, what it is, and what your kind of hopes are, I guess, for that?
Sure. So one of the ways that we try and study cancer out of the body is to use things like cell lines or other models. And there are really only two cell lines out there that we can use to study PMP. The problem with those is we tend to grow them in a flat glass or a plastic dish that doesn't really represent the environment that we find the PMP or the appendix cancer cells in. So what we want to do, and what we've been doing, is to take the patient samples, isolate tumour cells from those, and then grow those in a 3D environment that really recapitulates the key features of the disease. So things like a 3D environment around them made of extracellular matrix, but also including things like the mucus component that we know is a really major feature of these tumours.
So you've mentioned here that you're using patient samples. I'm wondering, because you've mentioned previously that this is a rare cancer, where are you getting these patient samples from, and how are you kind of compiling them in a sort of biobank, would be my question.
So we use the existing infrastructure in the MCRC, the biobank there. Omer and his team can identify the patients as they're coming through, and we can have as many as a couple of samples a week, even sometimes, because, you know, even though the tumour's rare, a lot of those patients are coming through the Christie Hospital and to Omer and his team. So we're getting the access to those patients fairly routinely.
Okay, and is this one of the main biobanks or the only biobank for PMP in this country?
So it is the largest dedicated biobanking project for appendix tumours probably anywhere in the world. And that's partly because in the NHS there's a centralisation to two hospitals. So as a hospital, we see more appendix tumours than pretty much any other hospital in the world.
That's incredible. And I guess looping back to this study that you're leading on, Pat, I'm just wondering, Omer, about if you could describe the kind of composition of the team and the European aspect side of that, and why it's important that we have those ongoing European collaborations at this level.
So when we set up this project, we did that with the help of Cancer Research UK Accelerator Award. And the idea behind the accelerator is to accelerate development of new treatments in rare tumours like this. And part of the reason why we were an attractive team was because we included a consortium of centres in the UK, Italy and Spain who could all collect from the same types of patients and do exactly the same type of work in the three countries so that we can increase the numbers of patients we could have. So over a 5-year period, our aim is to have had 400 to 600 appendix tumour samples, which we've highly characterised, for which we have models for that we can use in the lab.
Incredible. So coming back to you, Pat, in terms of this study, what would be your kind of top level of what you've achieved so far?
Okay, so far across the project, what we've been able to do is get a really deep understanding of these tumours, both the primary and the disseminated disease at a transcriptomic level and a genomic level. But in parallel with that, we've developed well over 100 models now, so mini tumours, organoids that we're growing in the lab that we can then use to understand what the environment is that these cells need to grow. But we can also then use them in future to try and understand which therapies might be useful for specific patients.
Incredible. And I guess you've kind of mentioned, Omer, that you'll have this vast number of samples over the next 5 years. My question to you, Pat, is what are you hoping to achieve throughout those samples within this time scale and then, beyond?
Yeah, so the real key for us is to take that information that comes from the transcriptomic and genomic studies and then translate that through to identify some therapies that we think might be useful that we can then test in the models. So that's what we're starting to do at the moment, and that's what we're looking to expand on over the next few years.
Amazing. And coming to you, Omer, my question would be, what do you hope the impact will be on PMP patients in the future from basically kind of using the outcomes of this research?
I think what we really hope is that there's going to be the hope of a new drug, a new treatment that we can give these patients. They haven't had anything to celebrate as a new option for 20, 30 years. And while surgery is a very effective treatment, it needs a lot of help. At least half of our patients recur. And it's really for those patients that we need to find a better answer to the treatment.
Incredible. So I think we've discussed kind of some challenges. I'm just wondering, coming to you first, Pat, what some of the key challenges within this research project are that you've had to overcome so far?
Yeah, so one thing that's quite unusual about this type of cancer is that the tumour cellularity is very low. So we get a patient sample, but the number of tumour cells can be very few within that. So dealing with that has been something that we've had to overcome. And working directly with histopathologists has been really important there because they can point us to the right samples and the right areas within a sample. And at the same time, developing the methodologies to more successfully grow these organoids by changing the composition of extracellular matrix or the chemical factors that we give to the cells. We can use those kind of methods to try and increase our chances of success.
Amazing. And I'll come to you to ask about some of the key challenges that you've experienced as well.
Yeah, so I think as Patrick alluded to, this work has required a huge number of people and a very big team, and it's required them all to be invested. So from the moment the patient is chosen for the surgery, their nurse specialist, for example, may speak to them about this project. We have surgeons who need to take samples as part of a protocol so that they can be collected and used for this research. We have theatre staff that then put them in media and pots that can then be used. We have pathologists then sampling them. We have other members of the research team transporting. We have people extracting DNA, RNA, and of course Patrick's team growing these in the lab. And then we have a huge number of people who've invested a significant proportion of their career studying and researching it. So I think the challenge has been to set up that team at a very difficult time in the pandemic, but we've come through that. And what we now have is a group that's very invested in finding a solution.
Incredible. So I think as my final question, we've covered the amazing work that you're doing in terms of this organoid development. I think coming to you, Omer, would be, we've talked about this one research area in PMP. What would be your hopes for the future of the research of PMP as a whole over the next 10 years?
I think that there are a number of aspects of PMP that need improvement. So we've talked specifically around drugs. There is the element of education and awareness. We haven't really been able to touch on screening, but through the work that we've done here, we may for the first time be able to do things like develop blood tests to pick up appendix tumours, for example. And what we really want to do is we want to involve patients in solving the problems that this disease brings. Survivorship is another area. Recurrence, for example, half of our patients will recur, and that will happen at a different phase in their lives. So I think what I hope is that we will have answers for people who are donating the samples today for their recurrences and their problems in years to come.
Thank you both so much for coming and speaking with me. It's been great to hear about appendix tumours and PMP, the Biobank, the CR-UK Accelerator Award and the organoid development that you're doing here in Manchester, and the fact that we're kind of leading on the progress for PMP patients and hopefully improving patient prognosis for kind of patients around the globe. So it's been wonderful to speak to you both. If any of you would like any content, there are lots of links down below from our website for you to read more about PMP and the work that Omer and his team are doing. So thanks for now. Bye.